Okay, I think we can make a start. So this is week number 9. We are going to discuss 2 topics, regression and also regularization. Okay, it's linked topics. Again, we will have quite a lot to go through in the first hour. And then in the second hour, I will start to discuss this highly variable gene and its implications in single-cell data analysis. This is a movement I believe I want to data science to teach biologists, not to rely on the differential expression analysis, but to look at more about the variability between genes and also genes between 2 conditions. So that's some topic we will discuss, we will cover in the next, the second half of this morning's lecture. So this morning we will go through regression methods and also to see how the lineage algebra will be used in solve this problem. Especially when we talk about the linear regression. Then we will look at numeric optimization problem, because that's a real underlying algorithm used when we use these packages. The package will not go through using lineage algebra, but using numerical optimization to find the solutions for your regression problems. Then I think that's a very important topic. What is regularization? There are a couple of related methodologies we can use to do this linear regression problem after established relationship between variables. Then want to control the numbers of input variables. When you have a lot, their method can lasso, for example. We need to have this concept established on your mind, so next time when you encounter these terms, you don't feel surprised. Okay, how many of you have never heard about lasso, L-A-S-S-O? Any idea who has what lasso is? You get some idea about lasso. Yeah, So you need to have this idea, and otherwise, when statisticians talk about this, they have no time to explain to you what lasso is. Like people will not have time to explain to you what PCA is. They just say, okay, we use a PCA to do the dimensionality reduction. Here, people may discuss with you, I will use lasso to do this regularization for your coefficients of your regression coefficients to find the most important features. So you need to have some idea of what they are talking about. So this is what we want to do. Okay, for linear regression, I don't know what's your impression before. You may say, okay, I saw this using Excel spreadsheet to solve the problem, so you can do this, and you can use R to do this fitting. Okay, I think this is MATLAB, but they always generate some tables, looks like this, give you the estimates of those values. So so the lecture here is to try to help you to fully understand what those are and how those are computed. So regression is a machine learning method. We separate the machine learning method into supervised and unsupervised. So clustering is unsupervised. Well, regression is supervised. Supervised means you need training data to help you to establish this relationship between variables. After that, you can use the build model to predict if you have a new variable input, and you generate the output of based on your model has been trained. So this is a general expression of this regression. So we have input of x. It will be, in our term, this data points. So we will have many vectors. Each vector is a data point. So we need to have these data points to predict another set of data points. So the y normally will be a single value. So if you have given 100 inputs, you will generate 100 numbers of y. So this y called the response variable. So normally n equals to 1d. So in the expression of this, you will see, okay, we want to establish or found a function f. This function will help you to predict the value of y to make this difference or loss function as small as possible. Okay, so you want to find a function f to establish the relationship between your input variable and output variable x and y, to make the loss function expectation, the average of that loss function value, as small as possible. So this is the expression in terms of minimization of this expected loss. So if you want to use least square expression, so this is a difference between so this is training data, all right? So you know what y should be when you gave x. So you want to establish a relationship of this function to make the difference between these two and also their norm, and this is second squared of norm, okay, as small as possible. So this is the expression writing in this loss function expression way. So we need to have very basic concepts in linear algebra in order to fully understand the next level of this expression of the variable relationships. We need to know dot product, okay? So this dot product has been everywhere, right? So dot product is everything. So I would see you have experienced a lot of examples. We simplified things into dot products. Here is the same. Another topic we need to know what's the norm of a vector. Okay, when you have a norm, you have a series of variables to describe your data points, and then the magnitude of this is x1 squared plus x2 squared to n, and then sum them and take square root. So this is called the L2 norm. Okay, let's express the regression problem into dot product. So this is not good. For instance, you have one point which is in here, okay? This one point is, so you want to establish the relationship between the change of your x. So this is like training data, okay? You know this is true, so you have a change of your x, so this is your x1, okay? And then you have, you know what change of your y1. So this point is x1, y1. If we assume this regression line goes through the origin, so we only need one parameter to establish this relationship. So we use one data point to train this model. That's enough, because the B, which the model we are going to build, contains only the one value, which is beta 1, C, okay? So this represents the slope, all right? So this beta 1, if I train this, it will be y1 divided by x1. So this is 1. So we need only use one parameter. To express this, so your data point, if you write into a matrix, so here is input data, okay? You have your input. Your input is x1. The reason I use a big one, big X, because I can have another data point, x2, if I have another point as your input, right? So you make a matrix, but in this case, we only have this. So to train this model, we just using B times X. If you don't know B, then you do the dot product with X, and you try to get Y. So if your input is only X1, then your Y will be just Y1, okay? So this is a dot product, because if you have another point, say, okay, X2, Y2, also on this straight line, you can express this into x2. And you can put this into here as well. Now, this big X becomes a vector if you do x1 or put in horizontal way, then you can get a vector out, y1, y2. So that's why we write in this more general ways to use big X, big Y, because if you have more training points, you will get more response for your training. Okay, so now we just assume everything is perfect on this straight line here, and there's no intercept as well. Okay, so that's why you can use this to express using dot products between vectors of your input to get a vector of output. So this is a dot product. Now the one I expressed here is slightly different because this contains a constant term. So even for this simplified version, if we allow the data to not go through the origin, then you need plus beta 0 to give you a term of constant, which this is a intercept, right? To allow you not only go through the origin, but if you have 2 points, have to go through a certain intercept. So this gave you the term to allow you to establish this relationship. Let's see. This y2 is somewhere here. So if that's the case, we need to have an expression to add one more to add 1 in the input variable in front of this, okay? Not from here. So for given every input point, you need to give a 1 here. So if you have xn in your input, you give a series of this. You just add one more dimension. So if you have single dimension, but you need additional 1, allow you to estimate the beta 0. So once you expand the dot product between x and the b, it becomes this formation. The b beta 0 times 1, so this becomes the constant. And then you have the first point, first dimension variable have b1 and the plus. If you have a b2, then you have multiple. So in this case, you have b2. In this example, it still have 1 point. So this is still one data point. So the expression is not so clear as here. And remember, this 1 and the 2 becomes 2D variable. So, okay. So if you have multiple time data, you will have I will have a matrix formation later, okay? So in this case, we just know you can express the straight line of the regression line in this form, okay? So for the norm, we have 2 different norms. Normally, we use L2 norms. This is definition. And if you want to find all the points which have the same magnitude, it will form this unit circle. Well, another norm is called L1 norm. L1 norm, if you want to find all the points which, this is the definition of L1 norm. Okay. So again, x1, x2 to xn stands for different variables. They're not different data points. Remember the matrix, you have points and you have variables to define the points. So the x1, x2 are the variables which this data is n dimension, so you just add them up. This is called L1 norm 2. So this is magnitude, this L1 magnitude. Now if you want to find all the points which share the same L1 norm, those points will be located in here. So this is if this is 1 and this is 1, 0, and this place will be 0.5, 0.5, okay? This point could be 0.3, 0.7. So all the dots in those lines share the same L1 norm. And when we talk about L2 norm, all the points share the same L2 norm will form a circle. Okay, so L1 norm, L2 norm gave you the data points share the same norm, a different shape of this to occupy the space. So we will use this concept when we do the regularization, and when we do L1 norm-based regularization, and we achieve lasso. And if you do L2, then you achieve elastic net. It's just different names, which underlines using two different norms. Okay, so this is a relationship between L1, L2 norms. Plot them onto the same figure, and then you can see they're different. Okay, some concept. When we talk about the regression like this, you use X to predict Y. So this is called simple linear regression because we only have one single variable. All right, so this is like a straightforward simple linear regression. But when you have multiple variables, again, so this x1, x2, xn are one data points, different variables. Okay, so you have more than one variable, and then in this case, you established a multiple linear regression. This is a simple linear regression example. You have engine size, which has input variable, just single one. Then your simple linear regression model will help you to predict the price of this vehicle, and you establish very simple regression relationship. This is an example of multiple linear regression. In this case, we have x1, x2, x3 represent different variables, engine size, horsepower, okay, lengthwise dimension. You put all the variables in order to predict the single output, y, which is a price in here. So this is a very typical multiple linear regression relationship model. Any questions so far? No problem. Okay, so this is right in the matrix way. Okay, I try to squeeze my notation in this corner, which is not enough. We need to have a huge space to illustrate this relationship, to translate this into dot product problem. Okay, so in this case, we have number one, this is a multiple linear regression expression. Okay, so how many dimensions we are talking about in this case. What's the number of dimension we are talking about? The number of dimension is a p, okay? So you need to look at this coordinates on your right. So this is x1, x2 until xp. So for each point, we have a p different values to define, okay? In the example of this will be the horsepower, the engine size, horsepower, length, width, height of the vehicle, and y is the price. So each row is one car, okay? And this is another car. So each car is defined by so many properties of the variable numbers. And you add one column of ones in order to let the model to return an intercept. Okay, this intercept are shared between all the samples. In this multivariable, you need a parameter of beta, which depends on how many dimensions we are talking about. We have a p dimension, so that's why you have a p beta 1 to p. You need to estimate those values plus the intercept. So you need beta 0. So this bold beta include a vector of coefficients of your regression. Well, this y is also a vector, depends on how many samples. In this example, you have n samples. So once you have all this write out, okay, those are the training data. Everything is known in this matrix. Then you can express your regression model in this dot product. So you have beta times this matrix and give you the result of y, okay? Plus, because not every point will locate it in the straight line perfectly, so you have some error to allow you to fit the model, okay? So our purpose is to find the best combinations of those beta values to make this error term as small as possible. Okay. So if you do the multiply dot product between this, so this is matrix times a vector. When you express them openly, so you will find the first row becomes this. Okay, so this is the model using all the parameters for the given data points to predict the y. This is sample 1, and this is sample 2, this is sample 3. So everything should be satisfied because you have an error term at the end. So when you see this simple expression, the multiplications between matrices, you know once you expand it, you get all the terms inside, look like this. Okay, so each row is a prediction model built on the regression relationship. And those values are shared, okay? Beta values are shared. So this is illustration for 2D examples. So we use variable factor 1 and variable 2 to explain variable y yield. So this will form not a straight line, but become a hyperplane. So you have this relationship, okay? So you use x1, x2, two variables to explain the y. So you have when you have a multiple linear regression, you have to think about hyperplane rather than just straight line, just one, okay? So two variables define the regression hyperplane, and then beta. This is for the variable 1, variable 2, and also intercept. So another place is we want to know there's multivariant. Okay, the multivariant, when we talk about the simple linear regression, multivariable regression, but when you see multivariant analysis, this will be very, very different from what we are talking. We are not going to talk anything about this, but you have to know once you see multivariant analysis, this means the output, the y, will be more than one, okay? More than one response variable. So your output becomes a matrix rather than vector. This is a completely different set of analysis. May have another book to talk about. So, but we normally will only consider the univariant. Okay, all we discuss for now is univariant. Your output is a vector. Your input could be one, could be simple, or could be multiple. So multiple and multivariant are very, very different terms. Okay. Again, there are some discussion about the difference: simple regression, multiple regression, and multivariant regression. Okay, let's come back to regression, linear regression expression using linear algebra. So I have an example that's like perfect points, and to train the model, 3 points in which located on this regression curve, regression line. If you look at every data point separately, you have 3 equations, okay? And assume beta 0 and beta 1 are unknown. So we can put them together in a matrix form, because beta 1, beta 0 are shared, so we only need 2 numbers. So this will be rewrite into a matrix or vector multiplication form. And then we simplify, put ones together, okay? All right, so this achieve at the end, that's the expression. So this is a real example to illustrate the huge matrix form of the expression, okay? So if you know this, you know this is correct. This form is correct. Okay? Any questions for this? Just ways of expression. Okay, this real data can show you that's true. So as you can see, we can use training data to establish the relationship, which is the y and x. This training established relationship, and this is a known. So we know this. We know this. What we want to estimate is the values inside here. So this is a question we want to solve. And if it's a simple multiplication, you can say, okay, b equals y divided by x. Then we solve the problem, right? Because this value is known, this is known, and this is a vector because we are talking about the linear multivariant, okay? Not multivariant, multivariable analysis. So we assume y is a vector. So we can do this, okay? In reality, the division, you cannot divide by a matrix because here is a matrix. What we need to do is to do inverse of the matrix, then times y. So this is the way we can directly compute the variables which we want fit, okay, the values of the model. This is known, so you can compute this beta two numbers very easily. And x, because you have already set the column with 1, so you can have beta intercept to be calculated as well. We have to compute the inverse of matrix. How to effectively compute this is a problem for the computational for computational computer science problem. Okay, how to solve this problem to give you very stable results, very accurate. So, but for the user, as a user, we can just borrow the built-in function in any scientific computing language. You should have the inverse function to give you this. Okay. So the definition of the inverse is for the matrix A, its inverse is once you multiply it by its inverse, then you get this I, which identity matrix. Okay, for the numbers, if you have a number n, then inverse is 1 over n. But in a matrix, you have this identity matrix divided by your inverse becomes the matrix itself. Okay, so this is an example. If you have two points, okay, if you have these 2 points, then you know your y should be this, and you can do this to directly compute the beta, okay? Beta value in this straight line. However, we don't expect they're perfectly fit values always. So there are always some errors when you put this line here. Some dots will be located on, some will be away. Our goal is try to find a straight line here and go through majority of these training data points and to make the difference, okay, between between real data points here, between this point and its expectation, the difference between this length, between these 2 points, as small as possible. Not consider only 1 point, but consider all this difference. All right? Look at here. So this is not a single point to the distance to the line. So it's not here, this point, you do a perpendicular line to find the distance. It's not. It's your expectation, which the distance as y, respect to the y, okay? Your observed to your expectation follow this, because this point, for example, if you have x and your y should be here. So that's the difference we are talking about, the d. So you want to minimize your predicted and the observed to allow you to give the best answer for. So this is formulate into optimization problem. If you haven't seen this before, this look a monster, but if you know, this is your real. So in this example, there are n samples, n data points. This is your observed point for one of the points from 1 to n. And then this part is your regression model to predict what y I should be, okay, based on this model beta, which, and also the slope and intercept for given x. So this is input. There are p variables, and this give you the expected y, and the difference between your observed and the predicted. So this whole term is a difference, which is just this, the yellow bars here, right? You have a lot of yellow bars from 1 to n, so you put them together and also squared, because some of the prediction is bigger than your observed, some is small, but if you do a power square and then everything become positive, you can use this as a distance and you put them together. So you basically add all the distance. You add all the distance of these yellow bars together. And the goal for the optimization is to try to find the best combinations of your beta, 0 to beta j, p, okay? So it depends on how many variables you want to consider. It's single or multiple variables. And you want to find these combinations of beta values to give you the total summation of this as small as possible. And the solution of these combinations of beta is your result of your optimization. So how you explain this? If you can explain this, majority of this optimization problem will be formulated in this way eventually. The difference will be inside here. What's the loss function may differ, but this whole term, whole framework looks something like this. So you are, if you become familiar with this, you can explain this in your language. You should be okay with a lot of this optimization problem, okay, later. So I will jump to the optimization solutions for this problem using numerical computation to just search for the space, just combinatory random search sometimes. They're random process. I will skip this analytical solution. So analytical solution is something like this. So we know if you have a perfect fit, or the term is the same on the lines, then you can easily compute this beta. But in reality, if we have imperfect points, then the solution will be this, okay? If your beta satisfies this relationship, you will get the minimized difference between your expectation and the explanation of your observation. So this is called the normal equation to give you the solution for beta. So if you rearrange your beta and it's become this. Okay, so this is a perfect situation beta. You can estimate using this, but in reality, the beta you will do some of the trick. Okay, you do X transpose. You know how have this big matrix you transpose and times itself. Okay, then you take inverse of this. And then you times X transpose times Y. This will give you the beta. Okay, there are some linear algebra theory, including to take a derivative of those expression. Okay, so the error term is expectation times itself. This is basically the squared, and you expand this, and you try to solve this problem at the end. The best solution will be the beta is this. Okay. So. This is an expression you can use to solve this problem using purely linear algebra operation. Matrix multiplication, inverse of matrix, and times this vector. So either way, so those are the linear algebra solution. This is called a normal equation, or this naive solution. Either way, we can try this in Thursday computation session, but that's the idea. Okay. This expression is so commonly used. So so it's so common. So this language, computation language like MATLAB and Julia, invent a new operators to replace this. So if you have a linear regression problem, you want to compute a beta, then what you can do just using this backslash. So they write in this way. So someone put this on their t-shirt to say, okay, MATLAB, the whole thing of this is this expression. Okay? Because it's so commonly used, that's why this language invented a new operator, which this divided by y, they gave you b. Okay? And Julia is so similar to MATLAB, so they also have this operation. You will see this result is the same as this. Remember, this is vector, this is a vector, this is a matrix. You want to get the beta combinations, the vector of your regression coefficient. So to use a regression model to estimate beta using input X. That's a relationship. But when you call regression function in most of the languages, they are not going to use this solution because this is computationally more expensive. Compared to what? Compared to optimization procedures. There are some other small details. When you compute the inverse of the matrix, there's a way called a pseudo inverse, which more stable computationally. It uses VIDI inside to do this. So you can see to do the inverse, you use the P inverse to get the same results as you use inverse. So this is a P inverse function. So you inverse this to get Y. So there are many, many different ways you can compute. You can compute this. Okay. You see here, the first method is just using this operation. Another is to do the inverse. So this is a normal equation. And you replace the inverse function using the pseudo inversion, you get the same results. Or you can do this more explicit way, which very similar to mathematical expression, you can still get the same. Or even you use this, you do the inverse times Y, you get the same results. Or if you use this, so all the expression I put here, you will give you the same beta value. So which one we are going to use? The limitation of this is sometimes not so stable, especially involved in this inverse operation. And it's computationally expensive. So we are going to do the optimization based on sum squared residual, and this is this W is a replacement with beta the same. So X times beta is a prediction of Y, and then you found the difference and square it and try to find the minimum when you have a different way to set up W. Okay, so this is a definition of the loss function. Loss function or cost function are the same. So this is a loss function terms. And the visual expression are those bars, the length of those bars. So you try to minimize the length of those errors. So optimization is a computational procedure to try to find the best estimate or combinations of estimates of parameters in your model. I explained this just like, I don't know what this called. It's a balancer or something in the musician field. You can have a lot of parameters to change. You have a combinations of those volume in different channels. And what I can do is to change those values, change those combinations, and to change the quality of sound or for some purpose. So my feedback from the audience, so audience say, okay, good, oh, terrible. So that's the loss function value I can get from the feedback. So I can make a change in this panel different parameters and ask audience, is it good? Oh, you can make it better, so I can change until I adjust all the combinations of parameter values at the end to maximize the satisfaction of the audience. That's the procedure of optimization. There's no difference between the process I just described and in terms of computation. What computation need to have the feedback function. The feedback function is the loss function, is the penalty function, the cost function, whatever you call, they are the same. As long as you have a cost function, the problem is half solved, because modern computation has so many different setups for optimization purpose. As long as you can define your problem using a cost function, your problem is almost solved. Okay, any of those machinery of optimization will help you to find the best solution, find the best combinations of your parameters to give you the best solution. So those rely on to find the minimized cost function, depends on which cost function you are using. In this regression problem is called least squared function. Least square is the difference between observation and expectation and the model explained it, and then the summation of the square. Okay. Sometimes you have objective function which give you the maximized values instead of minimized, but you just put a negative sign because the machine of optimization may only find the minimized values. So if you really want to achieve the maximized values, you can put a negative sign in your loss function and give to the minimizer, the optimization machine, to find the solution for you. So this is a procedure cycle in the numerical optimization. You have a starting initial guess of the parameters. You can set random numbers, depends on how many variables in your model. You can put 0.5 in all of them, and give the function to give you the first evaluation. So you have to have this function to determine how good, how bad the solution is. Okay, this is the center of the function, the loss function. If your random guess is good enough and satisfies your stop criteria, then you can stop, say, okay, the solution is good. Normally, it should not. Then you have to find the best parameter, change one of them, and see whether the result is better. It's not, then change another. So how you are going to change those parameters, follow what kind of schemas. This is the art of the optimization. There are so many different ways, depends on your input is linear, nonlinear, convex, or non-convex. So all the solutions may different, okay? There. Try to find the best minimized function value at the end. Try to find them as quick as possible. Try to find them very stable. Okay, not run on Tuesday, get this result. On Thursday, you get different results. And you want to be stable, and you want to find sometimes it's called a global minimum. So if you have a landscape, there are many, many local minimum spots. You may have a solution which trapped in one of the solution, and you can never jump out and find the real solution. So there are many, many different ways. So that's, you have a gradient-based search, direct search, there are algorithms, okay? Try to help you to find the minimum energy or loss function values when you have different combinations of parameters. So this is x1, x2. Okay, so you need to find the combinations of x1, x2, two parameters to give you the best lowest value in your cost function. okay? In MATLAB, there are built-in functions, for example, like this, called a minimum search. F minimum search, which you need a name of function. So this function help you to compute if you have a given x. So the x could be input data with multiple variables. Okay, so this is a vector. So for this vector, this function will compute a cost of this solution. So you have initial guess and give the function. As long as you have the function, the result will be the best combinations of parameters. Okay. You can have a search with a boundary, so you know your solution should be in between 0, 1. You put x1 0 and x2 1. So you will the function will search the values within this region. For more sophisticated applications, their optimization toolbox provide more functions using different algorithms and solve different problems and provide alternative optimized solutions. I don't find open source products like Python gave you the solution as good as this. So MATLAB has its own values in this case. Yeah, I was surprised in Python you can hardly find a function which is so easy to use like this. Okay, found minimum search. Okay, inside those optimization function, gradient descent is one of the algorithms that's used mostly. Descent means you want to find a path which go down, okay? Because you want to get the lowest cost in your function solution. So you want to go down, so that's what descent. Where gradient will tell you the slope and the direction. So which direction you want to search when you iterate your combinations of parameters. So what's the value of next you want to follow to try? The gradient will give you the direction of the search. In this example, you follow the gradient to lower your cost. So you follow this gradient until you reach to the minimum, and then the gradient will not change. It keeps the constant. Then you have no way to go down, and you don't want to go up, so you stop there. So this is basically the idea. So you need to find a way to compute the gradients. So gradients is your change of your value. So you change a little bit of your combinations of your beta or W. That's a parameter combinations. If you have a multiple dimension, you change or maybe one or multiple dimension, and then you compute the change of a loss function. So once you change you moved from your current solution a little bit, whether your cost function will lower a little bit. If that's good, then you move that direction. So it depends on the ratio, these gradients, you move either toward that direction or move to other direction. You compute your local gradients and find the lowest point to go for the next move. So you update the combinations of your solution you want to try next from a previous step by follow these gradients, and you can change this constant. So it's called learning rate to see, okay, I want to jump very far away. I gave you a bigger number, or it's is small, then I will try just move a little bit away from this current solution, and the steps is using small steps. So you see this differentiation, the symbol, rather than use change. They're same way, but this is just because you have a high dimension, you want to do the partial derivative from different directions. Yeah, so this is an expression of the gradient descent, and so that's open so this is called the pseudo code for the algorithm, and this is also just to show you inside what's going on. So most importantly, you have to compute this gradients, okay, from different directions. I think we need a 5 minutes break. Okay. Okay, I think we should continue. So this is a algorithm pseudocode for description of the gradient descent algorithm. So this is illustration about process a cartoon to indicate how the combinations of the two variables, so one is W and one is B, will be searched on this cost function value landscape. And in this landscape, we will reach the minimum when the combinations of these two values, when they reach to this plateau at the bottom of this valley. Okay, the color indicates how high the loss function is. So our initial guess could be the combinations for this position, okay? So B is -4 sometime, and then this -3, so to give you the value of this cost function somewhere in the 13.5. And then to compute this derivative from both terms, then you move downwards to here, and then the next iteration you give move to here, and then move until you reach to the lowest. Then there's no change, then you stop your search, and then to give the best combination solution, which W should be 0.5 some, and then the B should be -1, something like that. Okay. So the learning rate, to how far you jump each step, will also have be able to determine the solution, the quality of your solution. So if you jump too long each time, you have a large learning rate, you overshoot the complexity of your landscape. If you move too small steps, then you may trapped inside this local minimum rather than you can go found the global best solution. There are some links between the Newton's slope-based solutions. So some people discuss the combination of those. So there's overfitting problems. So you found the best combinations of your parameters to give you the best solution, but sometimes you use too many parameters. So this is the situation which is not as good, but this is maybe just enough. So in this case, if you use just two variables, okay, constant and also one variable to explain, compared to these two quadratic terms, and then this may have a better fit for your overall solution. And this is example of overfit. Although you explain all the observation, but you may incorporate a lot of noise inside your parameters. So we try to solve this problem, and especially, so this is not the best example for linear because there are quadratic terms here. But we talk about if you have a theta 2 and many different variables, and you don't want to overfit. Okay, overfit also have the problem in this discriminative analysis. You don't want to do the overfit to make your classifiers more sensitive. Okay, this is oversensitive in this case. So this overfitting problem will be addressed by the regularization procedures. Okay, so you do linear regression, multiple regression model. You found a solution of the beta, so you have n variables, you have found a lot of solution. The best solution for the training data is something like this. So the regularization procedure will help you to get rid of this, because the contribution of the second variable is too small, so we reset that into 0. Well, the third variable, the coefficient absolute value is big enough, so we keep it. And for this, too small, so it's not very useful. It contributes too little to the overall fitting, so we regularize it into 0. Okay, 0 again, and we keep the big ones. So this procedure, this process is called regularization. Okay, to trim off unnecessary contribution of the small regression coefficients in order to avoid the overfitting problem. So we need to find a more systemic way to see what's the cutoff we should use to get rid of those values, because one of the value could be 0.09. Would you get rid of it? Or 0.1? So the procedure we are going to use is called a lasso, or the other one. But this is a definition, and the lasso method was proposed in 1996 and then put in the very famous textbook in these elements of the statistical learning, and become popular. This is to solve the problem of the overfitting in the linear regression coefficients. It uses L1 norm constraint. Okay. Remember, L1 norm is the summation of absolute values. If you have a beta with 0, with 1, with 2, so you have a large number of coefficients. We are going to take absolute value of them plus them together. So give you a term, so it's t. So this term will be put into the cost function for optimization at the same time. So the optimization function will not only consider how much the combination of the parameter to allow you to predict the results accurately, and also will consider how big overall values you used inside your model. So this is a lasso cost function. Before, which term is extra? So this term is extra term. Well, they use W, so I use beta, so the same. So W0 plus W1. So L1 norm of your values will be plug in your loss function. So this loss function not only determined by how best your prediction is to give you minimized error, but also rely on how much you used non-zero values to give you a best model. So if you have a lot of small values here, 0.01, 0.05, and this add up will give you the bigger values which inside here. You make your model overall loss function less attractive, less more, okay? So you want to minimize the terms here. You only keep the most important ones. You only keep most important ones, not using cutoff, but just let the optimization function to find it for you. Then, because you only select the most important ones, this becomes small, okay? So this becomes small, your overall loss function will be small. But you cannot make it too small, because once you make this, you throw out a lot of important terms. Your model fitting for the observation will be worse, okay? So you need to have a balance between the observation you can explain the user model and also your total cost of your terms. So you balance themselves automatically through this search engine of the optimization to give you a solution. So lasso is only difference from the linear regression least squared cost function is with this addition term to add the L1 norm of your regression coefficients into the loss function in order to achieve the balance of numbers of coefficients and its predicted model power. All right. So this is another way to explain the L1 regularization term, which is absolute value of those values. Okay. No matter you write into theta, W, or beta, is all the same. It's just terms you use to explain your model. And that's fitting, okay, your prediction power. So you want to minimize these values to give you the solution. So the whole term is called least absolute shrinkage and selection operator. Okay, we just call it lasso. Okay. So this is an extra term and different from the standard linear regression cost function. This is called a rigid regression, which is very similar to lasso. The only difference is to use L2 norm as a term. So this is L2. So this is dot product, which is just squared. Okay, dot product is you transpose, and dot product is just squared of all the beta values. Okay, this is the whole thing is, which is beta if you look at this expression, it's very similar to lasso. The only difference is here is the value of your coefficient squared and put everything together. Rather than in lasso, it's Wi, which absolute values. Okay. So this expression tells you difference between L1 norm and L2 norm, and in the search space, and then the cutting solution will be this touching point. Okay, this is like just 2D results. There are a lot of solutions from the combinations. Okay, but if you have a constraint, see, beta beta 1, beta 2 has to be located within this box, then the only solution of these combinations with the constraint is this touching point. Well, for rigid regression, you can have a lot of solutions by combinations of beta 1, beta 2. But if your constraint is these two beta value cannot be too big, the L2 norm of them has to be inside this circle, then the best solution will be the touching point from this sphere to this overall solution. Okay? So this gives you some idea about the difference between these two. So this solution, lasso, will give you a lot of this one variable become 0 because this shape is really sharp with angles. If you think about high dimension, it's a hypercubit, maybe it's called. There are a lot of spikes in the shape. So once you touch the solutions, possible solutions, and it will cut a lot of other variables become 0. Well, this is the most soft, and then there are some small values still in there. People also combine L2 and L1 norm into the more compound constrained term. So this is like a balance between L1 and L2. This is called the elastic net. So there's no essential difference between these three, but just how you penalize the values of your beta. Use L1 or L2 or use combinations of the two. The framework of the optimization will be the same. So you have this form of the loss function. As long as you can define this using different ways, there doesn't matter. The optimization solution will help you to find these combinations. So this R package used to implement lasso algorithms and also Python version. And in MATLAB, we have a MATLAB version. Okay, so in the last 30 minutes, I will quickly go through this study, which to see whether single cell gene expression variability is an important description of function of genes. So we normally think the noise will not have anything to do with the function, because if you hear the noise, you don't think it means anything. But here in the single cell world, for example, we are considering a population of cells. They are homogeneous population. There's only one cell type, and all the cells look similar. Okay, there is no treatment. They all grow happily. So in this case, we see, okay, whether the gene expression in different cells. So gene, you can express, for example, this gene, if you think about it, one gene, this gene may express here, not expressed in this one, but expressed here, and expressed here. So looks like a random, okay? So whether a gene express more random, some genes may express high here, and this cell may be very high, and this cell a little bit high, but this cell may be high, really high. So some genes will looks more random than others. So our question is whether this level of randomness tell you some function of this gene for this homogeneous population of cells. So this is a proposed idea, which in a review paper, it's not a review paper, it's called prospective. So people just put the hypothesis in 2015. So variation is a function. It says maybe in a single cell, the difference of gene expression across cells is related to their gene function for the cells. But at that time, we don't have enough single cell data to test this hypothesis. But as you know, this technique became available 2016, okay? And then become popular, so we can have data. You can start to look at the genes and their variability expression across cells. Okay, we are talking about the roles, each gene, and some genes are more variable than others. So we test this using several different cell types. One is lymphoblastoid cell lines. So this is what it looks like. And then airway epithelium cell. So this epithelium cell. And I will mention another cell type, but now I'm not going to tell you what the cell type is. I will ask you, and what's your hypothesis about, okay? So first, using lymphoblastoid cell line as example, we first look at cells in different cell cycle phase. This is cell line, so there's no different cell types. They are just lymphoblastoid cell. They are B cell derived. So we have no cell type issues. So everything is homogeneous. And then look at the cells in the S phase and the G2 phase. What you can see, the overall expression is indicated by the color. So those cells looks yellower than G1 because the overall expression, the absolute value of mRNA concentration is higher in cells in S2 and the G2 phase. So we don't want to cell cycle to interfere in our expression analysis. So we just remove those cells, only keep cells in G1 phase for our analysis. We also remove mitochondrial genes and also make the library size to be very narrow, and all the cells have to be in there, and using different algorithms to do the visualization, and also manually select a group of cells which are the homogeneous. So we try to select a set of highly homogeneous population of cells in order to have a good estimation of gene expression noise. So our definition in the noise is gene expressed differently among cells, which in the same cell type in the same states. So we want to collect a set of genes, cells, very, very homogeneous. So this is a dimensionality reduction. Different method gave you different results. That's what we found. PCA gave you results look like this, and t-SNE and the FATE gave you, even though t-SNE looks like there's no structure, FATE will be more sensitive, still found those cells in different states. So we don't want to include those cells in our analysis. We only use cells which in the center of this cloud, which in t-SNE it could be the center here, okay? So the 3D, we also give you one more dimension to look at your cells. So there are other methods as well. This is called spectral method, okay? So we use a 3D FATE and look at the structure within the cell population of the cell lines, which is supposed to be homogeneous. Again, those are the G1 phase cells. And then we draw a circle and select only the cells located in the middle of this cloud. Okay, so this is a selected homogeneous population of cells. Then we start to look at the gene expression. So each row is a gene. And you can see this gene and this gene have a similar average expression. If you do average across those cells, okay, over 2,000 cells, they have similar average. This is the expression not showing as a color, but as the height of the bar. You don't see there are a lot of zeros here, okay? So a lot of zeros in this immunoglobulin heavy chain, okay? We know why, because there's semantic recombinations happen in there. But anyway, some of the cells in this homogeneous population express this gene into the level of 600. So those are the UMI counts. A lot of these cells expression levels are zeros, but on average, you reach a level which is similar to this ribosome protein. Well, in the ribosome protein case, almost all the cells, 100% express some levels. They are housekeeping genes, but the average is the same as this gene. So you can see these two genes in this homogeneous population achieve the similar level of average expression, but using very different ways. One is consistently expressed across all the cells, which another is more random or sparsely expressed in highly selected cells, but in the highly expressed way, more abundant. The level can reach to more than 600, but in the ribosome protein case, you never above 100 or 150. Okay. So what we can do is to compute values to estimate or quantify the variation. We use coefficient of variation, and for the ribosome protein subunit 11, and you get 0.43. It's a CV, which is a standard deviation divided by the mean, right? So their standard deviation is small, mean is the same, so you get a CV like this. Well, this gene have really high CV, indicate it has a much higher variability in expression estimated using the coefficient of variation. Now we plot these two genes. They have a similar mean, okay? So the x-axis are the same for the two. Well, they have a very different CV. One is high, one is low. So this is high, so this will be immunoglobulin. This is the subunit of the ribosome. And we also know the relationship between CV and the mean should be a straight line if you plot them into this log plot. Remember, this is mean is in the log scale, and also the CV is in the log scale. Okay, so this is a log. So we expect to have a straight line for the genes overall. Follow this. If we know most of them are the Poisson random variables, of the expression. So this is a regression. Okay, so we have a mean of a CV in the log scale. Each point, blue point, is a gene. So those are all the genes expressed in the lymphoblastoid cell line. We see every gene in the biology expressed have more variability than expectation. So the expectation is this straight line of the Poisson distribution. Okay, so no genes jump to this region. There's a boundary which, in theory, you could jump below, but in reality, real point, there's no gene can jump below that cutoff. So any biological genes estimation have some variability in there. Our question is how big the variability is. Some genes may be far away from expectation given the same average. Some gene may have very small variability for given the mean, right? So we want to measure how much it jump away from this curve, regression curve. So we also use a nonlinear linear least squares. So give you the regression curve looks like this, the green line. Okay. Because in the highly expressed part, there's some reduction of expected CV in this. So we need to use a nonlinear regression to better to get a better regression curve to be fair to those genes, okay, in the high end of the highly expressed. Then we measure the difference between observed CV, which for this gene here, okay, and its expectation CV. So if you have average level in this region, in this bin, you should have, for most of the genes, you should have the CV which on the straight line or on this green line or on average should be here. But then the difference between your expectation and observation CV is this difference. So yellow bar. So we call this residual CV. Residual means one minus another, right? So gene have a higher residual CV will indicate this gene is more noisy. Expression value is more noise in given population of the cell. So the regression we use for the green curve is using so-called linear, generalized linear model. So it allows you to have this transformation to get some nonlinear features in the function. This is what we did in MATLAB, so the link function and also to fit the generalized linear model. So this is for lymphoblastoid cell line. It's not very good fitting here, okay? So there's some problem with this fitting, but it worked. So we found the genes which above this orange regression line, which are example are these 4 genes. We also found some genes which under this line. So under this line means the CV is lower than expectation if we believe majority of the genes follow the regression curve. So look at their gene expression profile of those genes which above the line. So those are this gene, which is this gene, or TB, which is this, this is CCL1. So this red box included 4 genes. Variability are higher than expectation. So in contrast, these 2 genes are here. So this here, this here. First, let's look at this magnitude of expression. So the y-axis, the limit is 1,000 here. This is only 10. This is 35. This is 200, 1,005. So, so highly variable genes, once this gene expressed in some of the cells, the level, the real absolute value is really high in those cells. Well, this lowly variable genes have overall the expression, they tend to express in most of the cells, but the level is low, and the variability is low for sure. So our question is, why? What gene are these genes in different tissue, different cell types? So we found highly variable genes. Those are examples in the lymphoblastoid cell lines. And we found, okay, overall the same immunosystem related cytokine-mediated signaling pathway enriched. So these are our impression for lymphoblastoid cell lines. So those highly variable genes are related to the immunity and the defense of the response. So this is a plot for airway epithelium cell. You also found a lot of genes which are away from the regression line. So this has an indication they are highly variable genes. So those are the highly variable genes we found in the epithelium cells. So in this enrichment analysis for those genes, there are some chemical related, okay, the immune response related, but you can also find the collagen formation, you found extracellular matrix organization. So those are the function which specifically related to the epithelium cells. This is dermal fibroblast. Okay, so you also found the genes highly variable, and as a function, you found this highly variable genes in the fibroblasts in skin. They are, for example, the keratinization. Okay, so the structure development. So some of this function enrichment terms you specifically found in those cell types, and it really reflects their function. So those are the cell types we used. Those are the function we found. Okay, so you found epithelium cells with a collagen formation. You found fibroblasts with a keratinization. So you don't want to switch them. You don't want to have a keratinization in the airway epithelium cell. So that's not good, okay, for the cell. So there's very clear and the cell type specific function enrichment in those highly variable genes, which identified exclusively for them. So my question for you is, what is this cell type? If you have a regression curve looks like this, and there's no highly variable genes detectable. So this may be one, but the magnitude of difference of the residual CV very small. So in this cell type, there's almost no highly variable genes at all. So what this cell type is? If you haven't seen the slide, let me know. So this is a comparison between this, okay, the pattern difference. You see a lot of highly variable genes in those cell types, but not this one. So this is iPSC stem cells, especially induced from the pluripotent cell. So it's chemically induced from the mature cell types and reverse back to the stem cells from the human. What we can see from this example is there's no highly variable genes in stem cells and no enriched function. So why? Because the iPSC is undifferentiated. It has not been differentiated into the specific function cell type. So there's no function that has been registered. It shows as a pattern which no highly variable genes can be detected. There are empty region in this area. So if we believe highly variable genes also in this region are linked with their specific cell type's function, then we can predict if we have a cell type which in the middle of the transition. So neural stem cell, for example, is derived from iPSC, and in the middle of the transition into more matured cell types, neurons or oligodendrocytes, astrocytes, it should have intermediate steps patterns. So you have a lot of highly variable genes in those differentiated ones, and no highly variable genes in undifferentiated. And neural stem cells are half differentiated, so we expect to see this intermediary pattern. So indeed, so you start to see some of these highly variable genes jump out from the curve, but not many. And we also tried collagen sites. Okay, collagen sites is very specific and in epithelium cells in the bile duct, and we found a function which relate to the response to the steroids, hormones, corticosteroids, and those are the steroids hormones that stimulate the endocrine system. So one of the functions for this bile duct, and then have this sensor cells to sense the change of this corticosteroids is a part of the function of those bile ducts. So the function for collagen sites include those sensor to sense this. So from the analysis of the highly variable genes, you can start to see highly variable genes should be everywhere as long as the cells has differentiated into some specific function type. So if we do the annotation of function of highly variable genes, we may found the biological function of the cell type. Okay, so this view is very different from only consider the average change. Okay, so on Thursday, my postdoc will come here in the computer lab to do a lecture as a guest lecture. Xuan will conduct the lead you to go through the computer session. Okay. I will have another meeting to attend. I will miss, but Xuan, your teaching assistant, and the postdoc Salim in my lab will talk about the highly variable genes at the end to see if you have two samples, the same cell type. One is treated, another, for example, diseased and healthy, whether their highly variable gene change, whether we can use that information to detect the sample difference. Okay. All right, so this is all for this week. We talk about regression and use linear algebra to express those regression problems. And we talk about the optimization and lasso. I hope you have a concept of what lasso is. At the end, I will emphasize the highly variable genes analysis, which is very unique. We can do this in MATLAB. Okay, Shran will show you how to do this analysis in MATLAB by click. All right, see you on Thursday.